Welcome back to the deep dive. This is where we take really complex, vital science, the kind of stuff that shapes how we see the world, and, well, we break it down, make it clear, make it usable.
Exactly. The goal is to cut through all the noise out there, help you get a truly sophisticated grasp.
Be properly well informed. And today, wow, we are plunging into something that's absolutely core to our health every single day, but it's still, surprisingly misunderstood.
That's right. We're talking about the intricate dance between free radicals and antioxidants.
And let me tell you, the science we've looked at, it throws up some genuinely surprising stuff, things that really challenge what a lot of us thought we knew.
Think of this as your pathway to really mastering this info. We're not just scratching the surface.
No way.
We're going deep into the fundamental biochemistry, piece by piece, like the professionals do. The aim is for you to get beyond just definitions, to really understand the why and the how.
Get that real insight.
Exactly. A level of understanding that lets you see the bigger picture.
Okay, so the big question guiding us today is this. How do these incredibly tiny, fleeting molecules have such a profound impact on our health? What's the latest science telling us about their roles?
And crucially, what does this all mean for, how we can best protect ourselves?
Right. Okay, let's unpack this. So let's start right at the beginning. Basics first. What are these things, these free radicals? We hear the term all the time, right? Health blogs, product labels.
Everywhere.
But what does it actually mean biologically? Our sources define them pretty precisely as highly reactive molecular species with an unpaired electron. Okay, unpaired electron. Sounds kind of chem lab, doesn't it?
It does sound technical.
But imagine, a person who's just intensely restless, always looking for a partner, someone to complete them, settle them down. That restless energy.
That's a great analogy.
It's kind of like that molecule with the unpaired electron. It's inherently unstable. It's not happy. And it's desperately searching for a chemical partner, basically an electron, to find some peace, stability.
That restless individual idea, it nails their inherent instability and extreme reactivity. These things are incredibly fleeting.
Fleeting how?
We're talking lifespans that are just ridiculously short. Think nanoseconds to picoseconds. That's 10 to the minus 9, maybe 10 to the minus 12 seconds. Almost instant.
Wow.
And before they blink out of existence, they smash into another molecule nearby. And here's the thing. In their desperate bid for stability, they don't just politely take an electron. Oh, no.
No.
They either whip an electron for the molecule they hit, or they force one onto it. And this is where the trouble really ramps up. Because in doing that, they don't just fix themselves. They actually create a new radical out of the molecule they just messed with.
Ah, so they pass on the instability.
Exactly. It's like passing the buck, chemically speaking. They pass on their restlessness.
So it's not just one bad actor. It sounds more like dominoes falling, a chain reaction.
Precisely. That's exactly what we call it. A self-perpetuating chain reaction. Think about it. One radical, trying to get stable, destabilizes something else.
Which becomes a radical.
Which immediately becomes another radical. Yeah. And that one goes off and destabilizes another molecule, and it just keeps going. On and on.
Like that restless person analogy, they find a partner but somehow make them restless too.
Exactly like that. Sending them off to do the same thing. It's a cycle. Now, the main way this chain can actually stop or get quenched is if two radicals happen to bump right into each other.
Okay.
Only then can their unpaired electrons pair up, stabilize both. But, as you might guess, that's super rare.
Why? Because they're so short-lived.
That's a huge part of it. They're incredibly short half-life, and also their concentrations in our tissues are usually pretty low. So the chance of two of these fleeting low-concentration things finding each other in that tiny window of time, it's vanishingly small. Very unlikely.
Wow. Okay, so these incredibly short-lived, unstable molecules are constantly setting off these internal chain reactions all through our bodies. That really drives home the challenge here.
It does.
Which brings us straight to the impact. Why should you, listening right now, care deeply about these tiny, almost unmeasurable but relentless particles?
Because the damage isn't abstract.
Exactly. It's because their instability, this nonstop search for a partner, leads directly to real physical damage inside us. This isn't just textbook chemistry. It's fundamental to our health, our longevity.
You've nailed the connection. The biomedical importance, understanding the consequences. You just can't overstate it. When we look at the specific damage, we're talking about attacks on the very machinery that makes our cells work.
Like what specifically?
Free radicals broadly go after three main classes of biomolecules. Essential stuff. Our nucleic acids, that means our DNA, our genetic blueprint.
Okay, critical.
Our proteins, the workhorses that do almost everything in the cell. And the lipids, the fats that make up our cell membranes and circulate on blood at lipoproteins.
So membranes, proteins, DNA, basically everything.
Pretty much the core components. This widespread assault is the root problem, and it's what underlies the development of many serious diseases. Let's get specific, because this is where it hits home. First, cancer. When radicals hit the individual bases in our DNA, they cause chemical changes, basically altering the code.
Mutations.
Potential mutations, yes.
Yeah.
Now, our bodies have amazing repair crews constantly fixing this stuff, and this is a huge but, if those changes don't get repaired properly, they can be passed down when cells divide, inherited by daughter cells.
And that's how cancer can start.
That unrepaired, faulty DNA has the potential to initiate cancer, yes. And there's more. Those toxic dialdehydes we get from lipid peroxidation, the damage to fats, they can also directly mess with DNA bases, another risk layer. Then you've got atherosclerosis and coronary artery disease. Heart health. This pathway is really intricate and, frankly, fascinating. Radicals specifically modify the proteins or lipids in LDL, low-density lipoprotein.
The bad cholesterol.
That's the one. This radical damage changes LDL's structure. And the key consequence. This modified LDL is not recognized by the liver LDL receptors.
So the liver can't clear it out.
Exactly. Normally, your liver has these docking stations that grab LDL and pull it out of your blood, keeping cholesterol levels managed. But damaged LDL, the liver just doesn't see it. It flies under the radar.
So where does it go?
Instead, this abnormal LDL gets gobbled up by something called macrophage scavenger receptors. Macrophages are immune cells, part of the cleanup crew.
Okay.
But these macrophages get engorged with this modified cholesterol. They become these lipid-stuffed cells, and alarmingly, they then burrow under the lining of your blood vessels, the endothelium.
Especially if that lining is already a bit damaged.
Precisely. If there's pre-existing damage or inflammation, it's worse. And then, these overloaded macrophages actually die because the cholesterol levels inside them become toxic. And this whole cascade radical damage to LDL, macrophage uptake, infiltration, cell death that directly leads to atherosclerotic plaques, those hard, fatty deposits inside arteries.
The blockages.
Yeah, they narrow the arteries. And in bad cases, they can more or less completely occlude, block the vessel. That's what causes heart attacks or strokes, cutting off blood flow. And finally, let's touch on autoimmune diseases. Radicals play a sneaky role here too. The chemical modification of amino acids in our own body's proteins.
From radicals or those lipid breakdown products?
Both, yes.
Yeah.
Either direct hits or reactions with peroxidation products. This changes the proteins. Now, your immune system is brilliant in telling self from non-self, right? Invaders versus your own tissues.
Usually.
When these proteins get chemically altered by radicals, the immune system can get confused. It might suddenly see them as non-self.
Foreign.
And attack them. It mounts an attack, making antibodies against these altered self-proteins. But the real danger is that these antibodies may also cross-react with normal tissue proteins.
So they attack healthy stuff too.
If it looks similar enough to the damaged protein, yes. They attack healthy, undamaged tissues. And that friendly fire, the body attacking itself, that's how autoimmune diseases get started.
That's a genuinely chilling sequence of events, from one tiny electron messing up all the way to these major diseases. So given all this destructive power, this range of damage, what stands out to you? Which radicals are the absolute worst offenders in our bodies?
That's a key question.
Yeah.
And it brings us to a core concept.
Yeah.
The most damaging radicals in biological systems are, without a doubt, the oxygen radical.
Oxygen radical.
They're so important they get their own category, reactive oxygen species, or ROS. When we talk ROS, we mostly mean a trio of bad actors: superoxide, that's O2, hydroxyl, OOH, and perhydroxyl O2X.
The main one.
Those are the main ones. And the widespread tissue damage specifically caused by these oxygen radicals, it's so significant it gets its own name. Oxidative damage.
Oxidative damage, okay.
And logically, anything that protects us against this oxygen radical damage, those are our antioxidants. So when you hear oxidative stress or antioxidant benefits, it's all about this internal battle against these specific, highly reactive oxygen molecules.
That clarifies a lot. Oxidative damage from ROS fought by antioxidants. But if these radicals disappear in like picoseconds yet cause all this specific damage, how on earth do scientists measure the overall total body radical burden? How do we quantify this invisible fight inside us? It sounds impossible, like catching smoke.
You're right. Measuring the radicals themselves directly in a living system, pretty much impossible. Getting smoke is a good way to put it. But what we can measure is the evidence they leave behind, the footprints, the chemical debris from their attacks.
Yeah, the aftermath.
Exactly. Forensic chemistry, basically. One of the main ways is by measuring the products of lipid peroxidation, the damage to fats and membranes and lipoproteins we talked about.
Okay, how do they do that?
There are a few clever lab assays. One is the FOX assay, ferrous oxidation in xylanol orange. It measures lipid peroxides directly.
How does it work?
Well, under acidic conditions, these peroxides can oxidize ferrous iron, Fe2+, into ferric iron, Fe3+. This A3+ then reacts with a dye, xylanol orange, forming a colored compound, a chromophore.
So more color means more damage.
Exactly.
Yeah.
The intensity of the color tells you how many lipid peroxides were there. It quantifies the damage. Then there's TBRs, thiobarbituric acid reactive substances. This measures dialdehydes, like malondialdehyde, which are breakdown products of lipid peroxide.
Secondary evidence.
Sort of. These dialdehydes react with thiobarbituric acid to form a red fluorescent adduct. You measure the fluorescence. More fluorescence, more dialdehydes, therefore more initial lipid damage. Results are usually reported as total thiobarbituric acid reactive substances, another good metric.
Both giving a snapshot of past damage.
Right, and there's a really elegant non-invasive method, analyzing exhaled air.
Our breath. Seriously.
Testing. It's fascinating. When certain fats and 6 polyunsaturated fatty acids get peroxidized, they release a gas called pentane. And when N3 polyunsaturated fatty acids get hit, they release ethane. Both pentane and ethane are gases. They travel through your bloodstream and you breathe them out.
So you can measure pentane and ethane in someone's breath.
Exactly. And that gives you a non-invasive, almost real-time window into how much lipid peroxidation is happening in their body. These techniques, blood tests like FOX and TBRs or breath analysis, are crucial for researchers. They let us see the invisible battle, track the impact of diet, lifestyle, disease on our internal radical load.
That's absolutely incredible. Our breath telling tales about this internal war. Okay, so we know what they are, the damage they do, how we measure the fallout. But the big question remains, if they're so destructive, where do they actually come from? It's not like we're trying to make them, right?
Right.
And here's where it gets really interesting, maybe even a bit unsettling.
You've hit a critical point. No, we're not trying to make them, but generating free radicals is often just an unavoidable side effect of, well, living. Our bodies have multiple ways of producing these oxygen radicals. It's not one source. It's a whole array of mechanisms. Some external, but crucially, many are internal, fundamental to just being alive. Let's start with outside influences. First, ionizing radiation. Think X-rays from medical scans or UV radiation from the sun.
Everyday things.
Everyday things. When this high-energy radiation hits us, it can literally split water molecules in our cells, a process called lysis. And that directly creates hydroxyl radicals. Remember them, the really bad ones.
So sunshine creates radicals.
Yep. A dose of radical generation comes with your tan. Then there are transition metal ions, specific ones, copper, Q+, cobalt, CO2+, nickel 92+, iron, Fe2+. When these are free in the body, and free is the key word here, they can react non-enzymatically, just on their own, with oxygen or hydrogen peroxide.
And make hydroxyl radicals again.
Bingo. Leads straight to hydroxyl radicals. This is why your body works so hard to keep these metals locked up, not floating free. We'll come back to that. Next up, nitric oxide, NO. This was fascinating because NO is a radical itself. It has an unpaired electron.
But isn't NO important? Good for blood vessels?
Yeah, hugely important. It's vital for cell signaling. It's the endothelium-derived relaxation factor that helps regulate blood pressure. It's a good guy. But because it's a radical, it can react with superoxide, another radical.
Uh-oh.
Yeah. When NO meets superoxide, they form something nasty called peroxynitrite, highly unstable. Yeah. And peroxynitrite quickly decays to form hydroxyl radicals, the worst ones again. So even a beneficial molecule can, in the wrong circumstances, contribute to the problem. It's all about balance. Yeah. Moving inside the body now to physiological processes.
Huh.
The respiratory burst of activated macrophages.
Immune cells again.
Yep. This is a key part of your immune defense. Macrophages engulf invaders, like bacteria. When they get activated by an infection, they go into overdrive the respiratory burst.
What happens in the burst?
They suddenly start using way more glucose through a specific pathway called the pentose phosphate pathway to make loads of NADPH. They start consuming a lot more oxygen.
Why?
The whole point is to produce cytotoxic oxygen and halogen radicals that kill phagocytous microorganisms. They literally weaponize oxygen to destroy the bugs they've eaten.
So they intentionally make radicals.
They do. The key enzyme is respiratory burst oxidase, or NADPH oxidase. It takes oxygen and reduces it to superoxide, kicking off the radical storm. The reaction is NADPH plus 2O2 AADP plus 2 AGM plus 2H plus 2H plus way.
That's powerful stuff.
It is. And it's so fundamental that even with a mild infection, we see plasma markers of radical lipid damage shoot up significantly. It shows our own immune system doing its job is a major source of these damaging molecules.
Wow. Our defenders are also producers. But you mentioned something earlier about radicals being part of just existing. What's the biggest internal source?
Yes. This is probably the most surprising, most impactful source for most people. It's the constant, unavoidable leakage from our own energy factories, the mitochondrial and microsomal electron transport chains.
Where we make ATP, our powerhouses.
Exactly. Where most of the oxygen we breathe gets used to make energy, the very process of staying alive. The problem is the chemical reactions involved, oxidizing fuel molecules, they proceed in steps. And during these steps, unstable intermediates pop up, including things like flavin semiquinone radicals.
Okay.
Now, the system tries to control these, but they form oxygen radicals as transient intermediates. And because these reactions aren't perfectly 100% efficient, there's leakage. Radicals escape. It's not a flaw exactly, just an inherent feature of the chemistry of using oxygen so intensely.
How much leakage are we talking?
Okay, get ready for the aha moment. This is the stunner. Our sources state that some 3 to 5% of the daily consumption of 30 mole of oxygen by an adult human being is converted to singlet oxygen, hydrogen peroxide, and superoxide perhydroxyl and hydroxyl radicals, rather than undergoing complete reduction to water.
Wait, 3 to 5% of the oxygen we breathe just turns into radicals.
Instead of being fully used for energy, yes, a significant chunk accidentally becomes reactive oxygen species. And do the math. This results in a daily production of about 1.5 mole of reactive oxygen species inside an average adult.
1.5 moles every day, just from breathing and making energy.
Just from normal metabolism. That means that simply living makes your body a constant internal factory for free radicals. They're not just invaders. They're an intrinsic, unavoidable part of being an oxygen-breathing organism.
That's genuinely astounding. It totally reframes the picture.
Yeah.
Radicals aren't just external threats, they're part of us, generated constantly. Okay. But despite all this radiation, metals, our immune system, our own mitochondria churning them out, we're not just falling apart, right?
Yeah. Right. We're still here.
So this must mean our bodies are incredibly resilient. They must have evolved some amazing defense mechanisms, some kind of inner shield against this constant onslaught.
Absolutely. The evolutionary pressure has been immense.
Mm-hmm.
And the result is multiple, overlapping, sophisticated mechanisms of protection. It's really quite ingenious. One of the most basic strategies is preventing radical formation in the first place, especially from those metal ions. Remember how dangerous free iron and copper are?
Yeah, they trigger hydroxyl radical formation.
Exactly. But crucially, these ions are not normally free in solution in the WASI. They are meticulously managed, sequestered, bound up.
Oh.
They're either tightly bound within proteins where they act as necessary cofactors, their reactivity controlled, or they're bound to specific transport and storage proteins that render them unreactive.
Like what proteins?
For iron, you have transferrin for transport, ferritin, and hemosiderin for storage. For copper, there's ceruloplasmin. Other metals bind to metallothionein. This binding effectively neutralizes their ability to start radical reactions. And bonus, these transport proteins are big, too big to get filtered out by the kidneys. So this binding also prevents loss of metal ions in the urine. Keeps essential metals in, keeps them safe, elegant. Then we have our powerful enzymatic defenses, the body's dedicated cleanup crew. First line, superoxide dismutases, SODs.
They handle superoxide, the one from mitochondria and immune cells.
Precisely. SODs take that highly reactive superoxide and catalyze its reaction with protons to make oxygen and hydrogen peroxide. The reaction O2L plus 2H plus H2O2.
Turning a bad radical into something less bad?
Exactly. Hydrogen peroxide, H2O2, is still reactive, but much more manageable than superoxide. SODs are crucial first responders, and that leads perfectly to the next layer, catalase and peroxidases. Their job is to deal with the hydrogen peroxide that SODs just made.
Mop-up duty.
You got it. Catalase is incredibly fast. It breaks down hydrogen peroxide into harmless water and oxygen. 2H2O2 to 2H2O plus O2. Clean and efficient.
Where do these enzymes work?
Often, the enzymes making superoxide, plus the SODs, catalase, and other peroxidases are all packaged together in cellular compartments called peroxisomes. It's like having the cleanup crew stationed right next to the potential mess. Very strategic. Minimizes damage spread. Then there's glutathione peroxidase. This one tackles the lipid peroxides, the direct result of radical damage to fats in membranes and lipoproteins.
What does it do to them?
It reduces lipid peroxides to hydroxy fatty acids. Much less damaging. It effectively breaks that chain reaction of lipid peroxidation we talked about.
Okay.
And here's a key nutritional link. Glutathione peroxidase is a selenium-dependent enzyme. You need adequate selenium in your diet for this enzyme to work properly. No selenium, weaker antioxidant defense.
And the glutathione it uses up.
Gets recycled. The oxidized glutathione is reduced back to its active form by another enzyme, glutathione reductase, using NADPH. So the system keeps regenerating its defenses. Beyond the enzymes, we have a whole arsenal of non-enzymatic defenses. These are molecules that directly trap radicals, acting like sacrificial shields. Radical-trapping antioxidants.
Like vitamin E.
Vitamin E tocopherol is a classic example. It's lipid-soluble, so it works right inside those fatty membranes and lipoproteins. It reduces lipid peroxides, stopping the chain reaction there.
How does it work without becoming a problem itself?
Clever mechanism. When it reacts with a radical, it forms the tocopheroxyl radical. Now, technically it's a radical, yes, but it's relatively stable.
Why stable?
Because its unpaired electron can be spread out, delocalized over different parts of the molecule. This makes it way less reactive. And crucially, this tocopheroxyl radical persists long enough to undergo reduction back to tocopherol by reaction with vitamin C at the surface.
So vitamin C recycles vitamin E.
Exactly, like a tag team. Vitamin C at the water-lipid interface regenerates vitamin E so it can go back to work. And speaking of vitamin C, ascorbate, it's our key water-soluble radical trapper. Works in the watery parts of cells and in blood plasma. It reacts directly with radicals, forming its own relatively stable radicals that also persist long enough to be dealt with safely.
Does it get recycled too?
Yes. Its radical form, monodehydroascorbate, can be enzymatically reduced back to active ascorbate. Or two of its radical forms can react together to make one ascorbate and one dehydrosorbate. This regeneration is vital for both E and C to provide continuous protection. And don't forget others. Uric acid, surprisingly, is a potent water-soluble antioxidant in our blood.
Uric acid? Like from gout?
The very same. Also, the huge variety of polyphenols we get from plant foods, colorful fruits, veg, tea, cocoa, they are powerful water-soluble scavengers. Big dietary contribution.
And in the fatty parts, besides vitamin E?
We have ubiquinone, coenzyme Q10, and carotenes, like beta-carotene, both lipid-soluble, protecting membranes, and lipoproteins. So you see, it's this incredible multi-layered network. Enzymes, small molecules, water-soluble, lipid-soluble, all working together.
It really is amazing, such intricate defenses against this constant internal threat. Logically, you'd think, okay, some antioxidants are good, surely more must be better.
Right. If a little bit protects us, wouldn't a lot protect us even more? Maybe prevent everything. That's the logical assumption, isn't it? The one that drove a lot of thinking and a lot of supplement sales for decades.
Right.
But here's where things get really complex, even counterintuitive. What's fascinating and frankly critical to understand is that sometimes these good guys, especially when taken alone and in high doses, can actually backfire. They can have unexpected, even detrimental effects.
Backfire how?
This brings us right to what our sources call the antioxidant paradox.
The antioxidant paradox.
It's a paradigm shift, really.
Yeah.
It means that antioxidants, the very things we praise for protection, can, under certain conditions, act as pro-oxidants.
Pro-oxidants, meaning they cause oxidation.
They can promote oxidative damage, yes,
Yeah.
instead of preventing it. This fundamentally changes how we need to think about supplementation, especially with high-dose isolated compounds. Let's look at vitamin C again. We praised its antioxidant roles, right?
Yeah.
Neutralizing superoxide, hydroxyl radicals. Great stuff. But it can also act as a pro-oxidant.
Yeah.
For instance, it can react with oxygen to make superoxide. Or, if free copper ions are around, it can help generate those nasty hydroxyl radicals.
But does that happen normally?
Probably not significantly. These pro-oxidant actions seem to need relatively high concentrations of vitamin C. And concentrations like that are unlikely to be reached in tissues from normal food intake. Why? Because of our kidneys.
The renal threshold.
Exactly. Once your plasma level hits about 30 micromoles per liter, anything extra you take in above maybe 100, 120 milligram a day just gets peed out. So the body has a built-in safety valve that usually prevents vitamin C from reaching pro-oxidant levels from diet or moderate supplements. But now, beta-carotene. This is where the paradox really hit hard. For years, population studies, epidemiology suggested beta-carotene protect against lung cancer, other cancers.
I remember that. Everyone was taking it.
Millions were. Huge supplement market. Then came the 1990s and two major intervention trials, big randomized controlled trials, where they actually gave people beta-carotene supplements.
What did they find?
They found an increase in death from lung and other cancer among people who were given supplements of carotene.
An increase, not a decrease.
An increase. It was a massive shock, a total reversal of what was expected, a real game changer.
So what went wrong? Why?
The problem is context. Beta-carotene is a good radical trapper, but mainly under conditions of low partial pressure of oxygen, as in most tissues. That's why the population studies look good.
Right.
But at high partial pressures of oxygen, as in the lungs, precisely where they hoped it would prevent cancer, and especially in high concentrations like you get from supplements, carotene is an autocatalytic pro-oxidant.
Meaning it fuels its own damaging reactions?
Yes. It can actually initiate radical damage to lipids and proteins in that high oxygen lung environment. The very place it was supposed to protect, it could harm, especially at high doses, like a fire extinguisher somehow becoming flammable under high pressure.
Wow, same molecule, different outcome based on location and dose.
Exactly. And we see a depressingly similar story with vitamin E, specifically alpha-tocopherol. Again, population studies suggested protection against heart disease. Lots of people took high-dose supplements.
And the intervention trials?
A big meta-analysis of intervention trials with vitamin E shows increased mortality among those taking high-dose supplements.
Increased mortality again, not protection.
Correct. Another sobering result, echoing the beta-carotene findings, reinforcing this paradox. Now, these trials mostly used alpha-tocopherol. That's just one of 8 forms of vitamin E found in food. It's possible, the sources suggest, that the other vitamins present in foods, but not the supplements, may be important. Maybe the whole food complex works differently.
Could be. But what about the mechanism for alpha-tocopherol's paradoxical effect?
It's subtle. Vitamin E works by forming that relatively stable tocopheroxyl radical, remember? It persists long enough to be metabolized safely, usually.
Right, stability is key.
But, paradoxically, that stability also means that the radical also persists long enough to penetrate deeper into the lipoprotein, causing further radical damage, rather than interacting with a water-soluble antioxidant like Vit-C at the surface.
So instead of getting neutralized quickly at the surface, it lingers and dives deeper, potentially spreading damage inside the fat particle.
That seems to be the mechanism. The very property that makes it a good antioxidant in one sense, stability, allows it to potentially cause trouble in another sense if it's not immediately quenched by vitamin C. It's a very fine line.
That is genuinely mind-bending. So our attempts to megadose these single antioxidants might actually disrupt the natural balance and backfire. It makes you wonder, if radicals are so bad, why would the body even have any beneficial use for them? Why not just eliminate them all?
That's a brilliant question, and it leads us to a really sophisticated, relatively newer area of understanding. The essential role of radicals in cell signaling.
Radicals as messengers.
Precisely. Turns out, nitric oxide, NO, and other radicals aren't just damaging agents. They are important in cell signaling. They act as precise molecular messengers inside our cells, helping to orchestrate vital processes.
Like what?
Our sources specifically mention their role in signaling for programmed cell death, apoptosis, of cells that have suffered DNA and other damage.
Apoptosis. Cell suicide, right, for damaged cells.
Exactly. It's the body's crucial quality control. When a cell is damaged beyond repair, especially its DNA, or becomes dysfunctional, potentially cancerous, it's supposed to trigger apoptosis, eliminate itself safely for the good of the whole organism, gets rid of dangerous cells.
Okay, vital process. So how do antioxidants interfere?
Here's the potential danger highlighted by the paradox. It is likely that high concentrations of antioxidants, rather than protecting against tissue damage, may quench the signaling radicals.
So flooding the system with antioxidants might silence these essential self-destruct signals.
That's the concern. You're not just neutralizing the bad radicals causing random damage, you might also be neutralizing the good radicals carrying vital instructions. And if you quench the apoptosis signals,
The damaged cells don't die.
it may permit the continued survival of damaged cells, cells that should have been removed. And if damaged cells, particularly ones with DNA damage, survive and multiply, well, that's increasing rather than decreasing the risk of cancer development.
Wow. So trying too hard to protect might actually undermine a different, crucial protection mechanism.
It's a powerful modern perspective on why oversupplementation might not just be ineffective, but could potentially disrupt the body's own elegant, natural use of these molecules. It highlights that incredibly delicate balance. It's not simple addition, it's complex regulation. Understanding this really takes us towards that professional level insight.
What an incredible journey today. Truly profound. Let's try and weave all these threads together. We started with free radicals, highly reactive, unpaired electron molecules, constantly seeking stability.
And causing damage in the process.
relentless damage to DNA, proteins, lipids, contributing significantly to major diseases, cancer, atherosclerosis, heart disease, autoimmune conditions.
Big players in chronic disease.
Then we looked at where they come from. Not just external stuff like radiation, but fundamentally from inside us. Our immune system's necessary actions, and even more fundamentally, just the process of making energy in our mitochondria. Constant, unavoidable internal production.
An intrinsic part of being alive and breathing oxygen.
But thankfully, we're equipped. Our bodies have these amazing multi-layered defenses, sequestering metals to prevent ignition,
Smart preventative strategy.
deploying enzymatic cleanup crews, SOD, catalase, glutathione peroxidase,
A rapid response team.
and a whole host of non-enzymatic radical trappers, vitamins E and C working together, uric acid, plant polyphenols, CoQ10, carotenes.
Lipid-soluble, water-soluble, covering all the bases. An intricate shield.
But then the twist, the counterintuitive insight, the antioxidant paradox, where high doses of isolated antioxidants like beta-carotene in the lungs or perhaps alpha-tocopherol can act as pro-oxidants.
Flipping from protector to promoter of damage.
And maybe even more critically, potentially interfering with vital cell signaling, like the signals for apoptosis. Quenching those signals could let damaged cells survive, possibly increasing cancer risk. It blows the simple more is better idea right out of the water.
It really does. What we've gained is a much more nuanced, sophisticated view. Knowledge is always most powerful in context, especially in biology. This deep dive shows crystal clear that more is not always better.
Definitely not.
It highlights why critical thinking about health claims, especially around supplements, is just essential. The body isn't a simple equation. It's this incredibly intricate system of balances and feedbacks. Trying to force one part with mega doses, even of something good, can have ripple effects, unintended consequences.
Disrupting the natural balance.
Exactly. It's a reminder of the elegant, delicate dance of biochemistry inside us.
Oh.
And why understanding the whole system, the context, is the real path to mastery.
Absolutely. So, knowing what we understand now, this delicate, essential balance between radicals and antioxidants, what might this mean for how you personally approach supplements, your diet, maybe even your environment? What other questions does this deeper understanding spark for your own health journey, your own quest to be truly well-informed? Something to think about.
Definitely food for thought.
Thank you so much for joining us on this deep dive into the fascinating and, often surprising world of free radicals and antioxidants. We really hope you feel a bit more enlightened, definitely better informed, and maybe, just maybe, a lot more curious.
Hope so.
Until next time, keep exploring, keep asking questions, and keep diving deep into knowledge.